This is a podcast from the Nuffield Department of Medicine. Today we spoke to Dr. Sergi Padilla-Parra about his research on viral entry. Hi Sergi.
Hi.
How do viruses enter cells?
The majority of viruses enter the cell through a mechanism called endocytosis. Basically, this mechanism allows the cell to internalize nutrients, big proteins, or to control the level or the concentration of receptors in the membrane. So the virus actually is going to hijack this mechanism, to take advantage of this mechanism to get inside the cell, and then is going to be transported into vesicular trafficking and eventually fuse. So my lab basically studies with the use of advanced light microscopy these processes.
How can we block these entrance mechanisms?
The important thing here is to understand quantitatively the processes that the virus uses to get inside the cell. Basically, we should go from a virus-centric approach to the interplay between the host and the virus. Our lab, among other viruses, is studying HIV-1. And this is a nice example of how the virus triggers some cell mechanisms in order to internalize and eventually fuse. For a long time, it's been understood that the virus fuses into the plasma membrane, but the new techniques that we are developing in my lab, based on light microscopy, have helped to show that the virus might be getting inside through endocytosis. A better understanding of these processes will undoubtedly help to develop drugs, small molecules or peptides that keep the virus at bay.
How can we improve antiviral drugs?
What we need is to apply these new methods and have a better understanding of how the virus is triggering some key enzymatic players here. So the idea is to follow single events, but also understand at the same time how these cellular factors are being triggered, are being used by the virus to get inside the cell. If we can actually determine the activity or the abnormal activity of these factors, then we can develop novel strategies, novel targets for the drugs to be more effective.
The most important lines of research that are developed in the past 5 or 10 years?
I think, importantly, the application of novel light microscopy techniques, like real-time single virus tracking, in this case applied for HIV entry. And it was discovered that HIV might enter the cell through endocytosis.
Why does your line of research matter, and why should we put money into it?
There are two lines of research that are really well, or the foundation of these lines of research is really important. One is the virus-centric approach and techniques that allow, for instance, to study a structure using electron microscopy, cryo-EM, and another line of research that is more based on developing vaccines and studying how the immunological system reacts to HIV infection. I think our research fits really well and fills the gap between these two lines of research. We have accessibility to single events. We can now follow single viruses and understand how and when and under which conditions this fusion occurs. So I think that this is worth investing in.
How does your research fit into translational medicine within the department?
There are two different lines of research that are actually quite translational. First of all is taking what I do at the single virus or single cell level and apply that for high content imaging. The development of new techniques in high content imaging will help to screen new drugs, to block virus entry. And on the other hand, of course, if we focus on which are the factors that are actually allowing or helping the virus HIV-1 to enter the cell, then we can actually point at these targets and develop new drugs, new peptides, new vaccines that will help to block the virus before the process of entry.
Thank you, Sergi.
Thank you very much.